A very good evening, everyone, and once again, welcome to the Indian Biological Data Center presents the IBDC Seminar Series for 2026, and we are back again with the April 2026 webinar today. That is on data standards and ontology in biological curation for data exchange, integration, and analysis, featuring Kalpana from Intact EMBL-EBI. So before going into the webinar, I just wanted to clarify that about the agenda and the some housekeeping instructions for the webinar. For this webinar, we will go ahead with the welcome, and then we will talk about little bit about the IBDC and its resources. And then we introduce our speaker for today's webinar, and followed by the Q&A session and concluding remarks. And during this webinar, the microphones will be muted, and you can use the chat for the questions at the end of the webinar. And the webinar is being recorded. So I think you all know about IBDC and its webinar series, and you can find the previous recordings of all the webinars on IBDC YouTube channel. So do check out. And about IBDC, that as we all know that before IBDC, we really have to depend on the archival infrastructure of international repositories, and we don't have a place here in India which create the infrastructure for the sustainable archival of different data types. So basically with IBDC now we have a place where it's a national repository of India where all the data that is generated from biological sciences in India can be deposited, and in a manner which allow the fair metadata to be recorded along with the data, and which again support the digital sovereignty and interoperability with global databases. So we do share the data with the global databases, and we also maintain our own data within India. So this is a step towards the data-driven decisions and self-reliance in biological data. So very quickly, you all know that IBDC is the first national science repository for biological data in India. It is supported by the Government of India, and it's a joint project of Regional Center for Biotechnology, National Institute of Immunology, International Center for Genetic Engineering, Biotechnology, and National Informatics Center. It's housed at the NCR Biotech Cluster in RCB Faridabad. And we have a disaster recovery site at NIC Bhubaneswar. So whatever data which is archived at IBDC, in real time it is synced with another data backup site, and it is dedicated to the nation by Dr. Jitendra Singh in November 2022. So we have an infrastructure for the archival. We have Brahm high performance computing facility, which has a combined storage capacity of around 5.5 petabytes and a compute power of 961 teraflops. And because this infrastructure is there at IBDC, we allow different researchers from different institutions to use this computational facility for the data analysis of their research. And very briefly, that the mandate and the mission of IBDC. IBDC is the national infrastructure, and data from plants, agriculture, microbes, livestock, and humans, and every biological domain is again archived at IBDC, and we provide that the data disposition and is done with standardized metadata formats, and it is secure. Long-term preservation will be there at IBDC, and we will provide persistent identifiers, and we allow interoperability within different data portals. We support different national mission mode projects, and we allow users to use our computational resource for their research. So this will allow agriculture innovation, climate resilient crop development, faster and evidence-based diagnostic, improved livestock health and productivity, precision and personalized therapeutics. And in briefly, what we exactly do is, our main mission is the biological data archive, and we allow HPC facility to be used within the research community. We also support national mission projects. We do a lot of big data analytics, and we all provide all the user resources that is required by the users in data submission and accessing our different services. Along with that, we provide different trainings and data submission workshops and data analysis workshops also. So as we know that each data without metadata is of no use. So every different database that we have all uses the standardized metadata schemas, controlled vocabularies, and ontologies in the archiving of different data submission. And we follow the Biotech Pride guideline and the feed protocols. And when you submit here, IBDC allows you different access modes, and these are open access, managed access, and no access. So why this is important? We all know that data should be fair. It not only benefit you, but it also benefits science and national scientific infrastructure also. So we have to follow a fair guideline for data archival. So whenever we generate data, not just we have to publish our research, but we also take care or that we submit data to public repository so that it can be reused and come into the domain of fair data. So currently we have these 8 different databases for different data type submissions. And along with that, we have BioNode, which is basically a dashboard for biological data analysis. And these different mission mode projects are being submitted here, and we provide all the infrastructure and data archival support to these mission mode projects. So now, along with the archival and other supports, we continuously organize annual events, and we have a computational biology conference every year, and so you can look forward to participate in these conferences. So coming to today's presentation, we are delighted to welcome Kalpana. And she's a very highly experienced bioinformatics and scientific database curation professional with over 15 years of expertise in molecular interaction, genetic variant curation, biological data management. She was a senior database curator at Intact EMBL-EBI Cambridge, and she's also contributed immensely in Excelra Knowledge Solutions, BioBase, and Parthys Reverse Informatics, where she has worked on across different domains in genetic variant curation, clinical phenotyping, and therapeutic intervention, supporting different databases such as GoBiome and Ingenuity variant analysis. So we are very happy that she took time today and for this talk. So Kalpana, welcome to IBDC Webinar Series, and now you can share your screen so that we can hear you.
Oh, thank you, Sonia, and thank you for a very detailed introduction. I didn't expect this, but thank you for the kind introduction. And without wasting much time, I would like to share, share my screen. Give me a minute. Sorry, I think I'm supposed to share. Okay, now. Yes, I found the screen to share. Sorry. Take me a while. Hope you're seeing my screen in the slides, basically, right?
It's visible.
Yes. Okay. Thank you. And thank you for joining me today for the webinar titled Data Standards in Ontology and Biological Data Curation, which is aimed for data exchange, integration, and analysis. Well, I'm not part of EBA anymore. My recent association was until February 2020 this year, but I still take part in the curation activities of the main consortium, IMX. I'm trying to give an introduction to what I do, so that, I mean, the database itself, and so that we can reflect the ontologies, whatever knowledge that we discuss here in today is reflected. How is it reflected in IMX consortium, and how the curation process reflects the ontology? Okay, so IMEX is in short for International Molecular Exchange Consortium, where you have a group of open resources, curation resources, in and around mostly Europe, and you have one institute, I mean, obviously an EBI in UK, and you have few other institutes in USA as well. So they are open resources. They were curating molecular interactions data from experimental evidence coming from scientific publications. So the collaboration enabled them to follow a common curation model and to use common control vocabularies, and also to share the curation effort, so there's no redundancy in curation, and stick to the high accuracy and quality control measures. Basically, there's every curated article is reviewed by a second curator, and until it is agreed among the curators, it is not set for release. And then once the data is made public, the data can be downloaded in multiple formats, supporting for, I mean, to enable data exchange and analysis in whichever platform which you want to use. Okay, so coming to the main subject of ontology, hope everybody is familiar, but I would still like to touch upon the basics so that, we are still in line with what we are talking about. So what is ontology? I borrowed the definition from NIH. It defines ontology as set of concepts and categories in subject area or domain, uh, which shows the properties and the relations between them. So this allows the data standardization. That means there's an exchange that can be enabled with shared meaning, which is important for semantic interoperability. Literally, it means that whatever the data and vocabularies that we choose to use is accessible to both humans and data machines. And that is what Sonia has been explaining. I mean, while she was introducing to IBDC, she was mentioning a lot about FAIR, and the foundation of being fair is provided by the use of standard ontologies and vocabularies. Not only that, usage of these vocabularies and ontologies help us to dwell deeper, look for deeper biological insights when we look at from the data perspective. And yeah, you can also find a pattern just by looking at the data, or you can also test your hypothesis within defined systems. That's the advantage ontologies can provide. The common example ontology that we are familiar with is gene ontology. And maybe some of you are familiar about Uber anatomy ontology, or short for Uberon, cell line ontology, and molecular interactions ontology. So I'll be touching upon gene ontology and molecular interaction ontology. Obviously, for gene ontology, because the most commonly used, and people can reflect the background knowledge. I mean, you can also you will appreciate the background knowledge, background information on gene ontology and molecular interactions ontology, because that's going to be a reference ontology for reflecting the terms that we look in, I mean, the concepts that we come across in this webinar. And cell line ontology, personally, I have, I mean, we are not going to look into the detail, but I just wanted to say that I have made multiple requests, hundreds of requests for new terms in cell line ontology. Okay, yeah. Why is it? Okay, so now how is ontology related to curation practices? Well, again, I borrowed the idea from NIH, which everybody can agree with, the best practices in curation. It involves finding the right ontology for your curation project process, and also selecting the relevant terms. It might appear simple, selecting relevant terms, but no, because there will be a lot of options for you for each ontology, and there'll be a degree of granulation in it. So you have to choose and define your rationale behind choosing it. And then the third part is if you haven't found the ontology term of your interest, then naturally you will be led to create a new term or edit an existing term. Well, creating a new term happens because you didn't find the relevant ontology terminology of your interest or for your theme. And editing happens, or editing is recommended when you have additional evidences coming to an existing ontology term, and then it needs some slight modification. So these creation of new terms and editing happens when you are confident about the subject. You are an expert in the particular domain, and then you choose to decide whether the new term needs to be generated or not, or edited or not. So if you are not confident, and if it is you're not sure if there should be a new ontology term created, or is an existing ontology that you didn't understand, or the synonyms are not matching to your ontology term in your mind, then it's recommended to submit a request, which some expert will pick it up. I'll explain the detailed process, the collaborative spaces and everything in the upcoming slides. Okay, so now we'll start with finding an ontology. So there are options where you can, multiple options to be specific. You have the Open Biomedical Ontology Foundry, short for OBO Foundry, and then Ontology Lookup Services hosted at EMBL-EBI, and then BioPortal and OntoB. BioPortal, I think it allows code-based queries as well. But then we are going to look into the bit more detail in the OBO Foundry, because they are the pioneers in setting up the principles of what we currently use for building the modern ontology terms. And ontology lookup services from EBI, maybe just that I'm biased, being an ex-EBI member, but it also provides this Zuma portal, which I found it very useful, because I'm not an expert in every domain that I come across, okay? So if I'm looking for saline ontology, which I had tried, there'll be like a lot of synonyms, and the flavors that were mentioned in the publications were not uniform, and so I was not sure whether there has to be a new term or not. So then you need a search portal where you can follow your natural language, and that natural language is mapped to and the defined ontology terminology. So that gap is filled by Zuma. So I liked Zuma, so I'm going to explain that bit in detail. But you can also explore other ontology portals, bioportals, and OntoB, and but anyways, basically they are umbrella services to and give their best coverage to all the ontologies. Yeah. Okay, so Open Biomedical Ontologies Foundry, yeah, as I mentioned earlier, they are the pioneers in drafting the principles or foundation of modern ontology development. So they created these principles, which is like all the ontologies has to follow an open use and collaborative space or model. And then that also brings to the next stage, that they should also follow or insist to follow common format and standardized relations, so that the data exchange is quite naturalized. Okay, so on the technical front, they developed something called the OBO file format, which is the biology oriented language specifically designed for ontology building. And then, since it is in collaborative space, you attract more experts in the domain, and then your term request or whatever created, so that attracts multiple reviewings from the expert. And if there's an exchange in the feedback or discussions, and then it gets validated. And for an existing ontology, with the availability of multiple evidences, with every day you get new research and new data, new evidence coming up, so then there might be some need to edit, and so that gives an versioning of an existing ontology term. So there's an option wherein you can go and see the history of the ontology, the life cycle, and then and then sticking to the textual definitions. But textual definitions, like saline ontology should stick to only it should get only it should carry only this the ontologies, this same ontology related terms, but not any, non-ontology related terminology should be never be included. So they make sure that, and so that it appears all the ontologies are, clean without any junk. And then, so I'm taking you to the space, the web portal. You can also explore in the Obo Foundry link. It's the same link, actually. So yeah, this Foundry invites you to find, use, and contribute to community ontologies. And you can search in the table and can use the drop-down menu options provided here. And it also provides what is called as the star rating for each ontology, and this is rated by the community uses. Yeah, so it helps us to narrow down which ontology for you to choose. But Obo Foundry has a good collection of science and biology related ontologies under their umbrella. Okay, so the collaborative space, as I mentioned earlier, is provided by GitHub, and this is the GitHub link for the Obo Foundry. So on looking for ontologies, there might be cases that you may not have found them in the published, released version of the ontologies. There might be cases where they are still under discussion, and you can also explore the GitHub space for issues that are open, and then you can see you can look for whether your terminology of interest is under discussion. There'll be like a lot of discussions going on, and you can see if, there are like 159 issues when I took the screenshot. And so you can go through them. You can basically query here. There's a query space. Try to use your natural language, or you can just rephrase it and edit it to see if your interesting ontology is somewhere in the pipeline. And yeah, coming to the EBI space, ontology lookup services, this setup is similar to Obo Foundry. Basically, I think they follow the same setup, the generation of file format and, collection of the, being the umbrella services of the ontology. Maybe it's just that it is hosted in EBI. Okay, so what is interesting to me is the Zuma portal, which is automatically used to annotate properties. What I mentioned is the gap between the assumption of the ontology term in our head and what is really mapped as ontology term by ontologists, the subject matter experts. There is always a gap, and I have noticed that. There will be bit of discrepancies, because when we were trying to map ontologies, there were cases where the ontologies had to be split. The concepts were multifolded, so you have to select two ontologies. So you should know whether you have to go for a second ontology, or a single ontology is able to cover all your terminologies, all the concept in the single term. So for that, yeah, Zuma portal is really help you. I wanted to give a demo, but actually it is showing an error today. It happens with me every time when I choose to give a demo, something happens. But I'm sharing you the link, and in the interest of time, at the end of the webinar, you can also feel free to explore it. And in case of any queries, you can mail to them. They'll be happy to help. Okay, so selecting the terms, because I'm not giving an example straight away, because I'm not sure which ontology to select basically, but sometimes in my case, I have used multiple ontologies based on the level of complexity. So based on the subject, if you're an expert, you'll get to know. It comes by experience. Okay, there's no hardcore rule to follow which ontology of your interest or not. And if you have excluded all the options, if you're not able to find them, then you have to create them, and then that comes to the experts in the subject. But if you want to, request for new terminologies also, there are like a list of things that you need to know before requesting. And these terms are needed even for creating new terminologies. Okay, so it's common. So basically we will be going to the GitHub space. For every ontology, we'll have the GitHub space. This is mandatory, because every ontology is made to be open, and it should be collaborative work from the community members. Okay, so then for requesting for new terms, so you should have the term name and then a textual definition and uh, definition references. Basically, I usually provide PubMed identifiers, which are good enough, and provide as much as synonyms possible because this helps us to avoid the risk of having duplicates within the system, which is a pain to clear them if gone unnoticed at the very early stage. And then to provide the logical structure, because when I say ontology, they are not just concepts, but also bring the relationship between the concepts. So then you have to pick and present the logical, I mean, choose the hierarchy in where it belongs to. I'll show examples in the later slides. So this is a logical structure. Essentially, you say that it belongs to this class and there's a relationship, maybe it's a child term or you need to create a new term for whatever reasons. And then once you provide or once you submit with these technologies in a GitHub as a ticket, and then so then it attracts, subject matter experts, and then there's a space for discussion with the community. And then once everything is sorted out, people agree to be created with a new ontology term. Then we have an Protege as an editor, which is an open source editor supported by Stanford University. And so you provide all these informations like the term, the textual definitions, and then synonyms, the logical structure, and then you get a file generated in multiple formats like OBO file, which is is the most recommended or essential format for the Foundry compliance, and then an RDF or all format. So, but anyways, these file formats are these are the common formats used, and the data, I mean, any format can be converted into another, so you don't have to bother much about it. And there's also every ontology term will have an unique identifier, or internationalized resource identifier, which is automatically assigned. For example, it will have a structure similar to something like, HTTP, like the OBO library, and under OBO Foundry, it is the Go ontology, and then the numerals, the rules that every ontology follows specific for itself. I'm going to take you to the IMEX consortium because this is where we are going to see how whatever the concepts that we have seen looked into so far is reflected. Okay. So for that, I have to give an introduction to the curation model. Bear with me. I'm not going to take much time. So as I mentioned earlier, so IMEX is interested in the molecular interactions data from experimental evidence provided in the scientific literature. So once a piece of literature is qualified to be curated under IMEX, the curators has to look into the details in the sense at the degree of, what is the type of interaction that is is detected, whether the participants have a physical evidence or direct or they are just proximal partners. And then you have to look for the experimental details. What are the biological rules? Whether there's a bait or a prey or an enzyme or an enzyme target, could be a competitor or a neutral component, whatever. And then if it is a protein construct, whether it carries any clinical variant or a mutation or a post-translation modification or any of these changes in the sequences, do they have an impact on the interaction strength? If yes, then what is the degree of the strength, whether they break the interaction or whether they just reduce the strength of the interaction or increase the strength of the interaction, whatever. So in that lines, then the space to build what I call this contextual interactome. This is essentially the from where the interaction has been detected, the host organisms. So it gives us a space to accumulate evidences on a particular theme on a tissue, human interactions detected in brain tissue. Like you can have multiple publications having interactions detected in that, and you can have one single data set on it. And not related to ontology, but the process to maintain the data standards, the protein updates and mutation update, because essentially we follow UniProt as a reference database for proteins. And then apart from sysprot entries, there are many entries which are still not fully reviewed, and there might be change in the sequences, the amino acid position. So this has to be reflected within the pipeline. So this and yeah. And then anyone who's interested to get to know the complete details of the curation process, well, it's up to you to choose, make a choice. But anyways, while I was in Intact, like my manager made sure that I don't look into the manual for at least for 6 months because it's too much of information and he didn't want me to be overwhelmed with the knowledge. But but no harm in going through the manual. So it is available in the IMEX consortium portal in the curation space. Okay, so the ontology structure is, as I mentioned, the curation model is deep as deep as possible, and so you have 1600 classes, and you have so many levels, and these are how are related, and it is hosted in the European Bioinformatics Institute OLS page. And I'm just highlighting, the interactor type because these are the relations, how the relations are being tagged. Then I want to tell you what are the data standards of the biological entities that we use. So the interactor type, we have 47 interactor type, and they are organized in their own level hierarchy. They have their own hierarchy under this class. So all the proteins are referenced to uniprot, even peptides are also referenced to uniprot. Genes have an ensemble, microRNA have RNA central, and small molecules are supported by KEBI, and complexes have complex portal identifiers. And all of them will be made sure they are up to date. Yeah. Okay, so this is our GitHub page, and even in this, in this page, you can also see the issue tracker that will take you to the GitHub space here, and this is the PSI control vocabulary GitHub space, and see we are also 4-star, got 4-star ratings. Okay, so I'm showing some examples of how the requested new terms requested from a curator. Hope you can all see. Pardon me for being, this the size is not maybe very appropriate, but you can also choose the link to go and see the entire conversation. So this is a term request by our expert curator, Sandra, and she wanted a turbo ID, and she has given which is a feature tag, and it is, she has provided the definition and the supportive public PubMed identifiers, and which also has another synonym. And sorry, I think it's a second request. Okay, so then where are these? So this come under the parent biotin ligase tag, maybe MI child of beta, which has the MI identifier. I'll take you to that part later. So then it attracts, develop a conversation, and you have the whole conversation history happening in this GitHub space. And then once agreed by everybody, then it is taken, you have the file generated, and it is picked up by the OLS in EBI. I think the processing time is at least 2 weeks or maybe less than 2 weeks to get it reflected in the OLS space, in the molecular interaction space. So as you can see, the turbo ID tag is a child term of biotin ligase tag. You have the relationship established. It is so you have the class, the major parental class, which is the molecular interaction. Under it is a feature type, and it is an experimental feature, and it's a tag feature. And under the tag feature, you have a biotin ligase tag is one of the feature type, and it is a child term of it. So all these well defined, and you have the definition and the exact synonym, how it will appear in our curation tool. The synonym reflects it. And then and this is the important part. This is the identifier. MI identifier, which has the 2433 as its identifier, and this has a specific format. This is the internationalized, identifier that is automatically assigned while you are using Protégé as your editor, because this is what your machine will understand as a turbo ID tag, and we understand the turbo ID tag as turbo ID tag, and the definition is all stored in the OLS. I have given the link for you to explore further. Similarly, well, I have also made requests, but my requests were very simple. I've just given the definition and where it fits and the reference ID, and yeah, my developer picked it up, and yeah, you also. This is another feature type. Yeah. Okay, so how is it reflected in building the interaction evidences? Well, the what you see in the left is the GPCR RAMP interactome heat map, and yeah, it was not, there was not an Excel. Of course, it was an image data, and I had tough time in decoding it. I had used AI tools to convert it into text, but then the accuracy, I have to check it again, so there's no point. So this beautiful, but it's very relevant data, very exciting data, for very tempting data for me to curate as well. So generated by Thomas Sankmar and their lab members. So with the detailed, curation ontology structure, so we were able to find out the relationship between we were able to reflect exact relationship that was studied in the literature using your ontology and using a curator tool. And this is what you see in here. This is a network actually which has been generated after the data has been released in public in Intact Cytoscape app. And as you can see, the interactors, the ramp proteins, the flavors, ramp1, ramp2, and ramp3, they are having interactors, and some of them are common interactors. And you can also see the edges. The circles that you see are the biological entities. Here in this case are proteins, and then the relationships are the interaction evidences. Here they are one-on-one interactors, like each protein was tested against every RAM protein. So it's one-on-one interaction. And so every line is an interaction evidence. And then you can also see there are few lines which are having higher color intensity than the rest of them, because some of these evidences were tested again, same binary pair tested again as a part of validation process by the authors. So it has multiple evidences. So the more the evidence, the more the intensity of the evidence. So this is the outcome of properly structured ontology-based curation practice. And this is how you brilliantly transfer the information from in a non-readable format, non-machine readable format, into something which is readable by both machines and human beings, and it is available for analysis. And this is a good ontology outcome. But then the benefits of ontology doesn't stop here. It can also allow you to look into more details, because just from the visualization of this network, you can see there are proteins and there are interactors and there are common interactors. And beyond that, there are some interactions having more strength. Beyond that, there is nothing much information that it can provide. But then you can subject this information, because it has all the data standards. All the RAM proteins have their identifiers, their relationship mentioned. Then you can subject this to any enrichment portal for Go on. Basically, people are familiar with Go enrichment in spaces. And so you can do some enrichment analysis or subject it to other analysis to get more biological insight from this data. So I did sample molecular function enrichment. And so I used this bit portal, and basically I gave the identifiers, the protein identifiers, the UniProt identifiers that we use. And then asked it to enrich for molecular function. I didn't care about the statistics or the degree of significance, because I was just exploring, just want to see what it is. And so, as you can see, you got the molecular functions, and then you can see the UniProt identifiers, and you have the heat maps and tells the degree of significance for each proteins. I just cropped the image, and there's a list of proteins. You can also try to do the enrichment in Klugo portal or any other portal that you're convenient to look into. Now let us see how because this is a Go ontology molecular function aspect. You can also let us delve into a bit more deeper part of like how Go gets this information. Well, this is again, I did a query using one of the UniProt identifiers highlighted here. And then I just made a query in Go, Quick Go version, which is again hosted at EBI. And I filtered for molecular function. As you can see, this UniProt identifier is for the RAMP1 protein, and you have the Go term function, molecular function, which is like, the calcitonin gene-related peptide receptor activity supported by the evidence code. And then that piece of evidence is found in the PubMed identifier. And this activity is found to, this RAMP1 protein is having a calcitonin gene-related peptide receptor activity on another protein. Well, the symbol of the protein is not here, but you can also, you can just click on it using the link provided, and there's a link and it takes you to the UniProt space to look into the protein details. So this piece of annotation is provided by UniProt, and then again you have two more pieces of the same activity coming from two different publications, again coming from UniProt, and also there are evidences coming from Intact, but mostly they are restricted to protein binding because essentially protein-protein interactions can not give much more contribution to Go molecular function seriously. So I have provided you the link. You can go and explore. So how does Go? Okay. So anyways, but still what we saw here was a G protein coupled receptor activity. But what we see here is the calcitonin gene related peptide receptor activity. So how are they related? Well, the details give the relation. So actually what you see here is the calcitonin gene receptor peptide receptor activity is actually a child term of G protein coupled peptide receptor activity. So in some enrichment applications, you have the option to restrict to the parent terms, then the restriction will happen at the, at this stage. But then you can also, there are applications that allow you to go to the more granular level of annotation, but that is for another session. Yeah, so so you can see how they are all related. There is there's a relationship, and then there are different color codes, and Go tells you that what does the different color code means actually. Can explore that for details. So Go basically, it was actually started during, when the gene sequencing projects of the model organisms that were completed in late 1990s and early 2000. So we had the yeast and the C. elegans and the Drosophila melanogaster full sequencing completed before 2000. And then accumulation of these sequences, molecular sequences, looking at that, it came to allowed to reach to an acknowledgement that there could be just a single limited universe of genes and proteins, many of which may be conserved in living organisms. So then this formed the foundation of building the gene ontology. So they compared the eukaryotic genomes, yes, as the yeast and worm, and then the large fraction, they found that large fraction of these genes displayed evidence of orthology. What is orthology? Orthology is their evolutionary relationship between a pair. Just say that they share a common ancestor. So this is like the 12% of the yeast genomes where putative orthologs in yeast, worm genes, were putative orthologs in yeast, which is kind of like 27% of the yeast genes. And most of these proteins have found a role in core biological processes such as, DNA replication, transcription, and metabolism. So then the three-way comparison among the budding yeast, worm, and fruit fly shows us the relationship can be extended. So then that's how this information laid the foundation of Go ontology, which they wanted to build the dynamic control vocabulary that can be applied to all eukaryotes as a knowledge of gene and protein roles in cells, and it is still accumulating the knowledge. So you have three aspects in gene ontology: the the biological process, molecular function, cellular component. We saw the enrichment of molecular function, and anyone can run the analysis. There are web portals that just allows you just give the gene names and look for you can pick whether you want to enrich the set of gene enrichment for biological process or molecular function or cellular component. It can be done. And if you look at the structure of how this terminology has been structured. So it has this accession number that is specific, the format is specific for each ontology, and you have the name, and it is the ontology class molecular function, and you have the synonyms. There are no alternative IDs, and then there's a clear definition provided by a PubMed identifier, and then you have a link to see the history, so that the versions. And then, yeah. So I think, I think we have covered the ontology. We have seen in reference database, and we reflected the use of ontologies for data exchange and transfer. And so with that, I like to come to the end of the webinar by giving the summary. So yes, ontologies are always work in progress. But still they allow the data to be available for both people and machines, as they aim to represent the current state of knowledge in biology. Obviously, it is constantly being revised and expanded with the availability of new knowledge. But it also means that the resources that use the ontology need to keep a track on the changes in the terms, which might be challenging, but the benefit is more weighing, because you'll be able to allow the data to be available for the greater part in accessing for both man and human, and it's available for analysis, and more it can provide more biological insights. Then it's more, I mean, the advantage is more than the challenges. Yeah, okay. So with that, I conclude, and I thank you all for patiently listening to me. I know this subject is a bit heavy. I tried my best to accommodate everything, as possible, cover the basics and given reference database as example as possible. That I stop, and thank you for listening, and let's open the floor for questions. I'm happy to take questions from you. I'll stop sharing my screen now. Yeah.
Thank you so much, Kalpana, and it's been a very nice presentation. So we have a couple of questions, and the first question is from Dinesh, and he wanted to know that there are duplicate gene ontology terms in molecular function, cellular component, and biological processes. This is confusing when we cluster them into the same ontology terms, and many times we need to manually look into this, and again cannot done several terms and genes. So any comments and suggestions?
So, I mean, there will be always an overlap in how the terminologies are, structured, because sometimes it can have multiple parents. And so based on that, the relationship between the biological process and molecular function will be, there will be divergent. But I bet I mean, nobody is going to harm you for choosing one side. Okay, so these are understandable. People agree that, okay, and you just have to give your own reasoning, because why did I choose? Or maybe you can just say that I couldn't decide on this, and you can also invite the experts to make a comment on it. But in my opinion, while I was using ontologies for mapping, whenever I had come across different concepts, I chose my rationale behind it, and I just documented my reasoning behind choosing the ontology term for the use. Hope it made sense to you. Yeah.
And another question is, could you please explain how gene ontology analysis can be applied to understand the functional roles of genes and identify critical pathways or biomarkers involved in breast cancer progression and treatment? I think it is.
That's a detailed process. I mean, that we need to do some enrichment analysis. For beginners, I would suggest you to, I mean, there are platforms. Basically, you start with the list of genes. You need to have unique identifiers, the gene list of genes or proteins, whatever. And then you have to load it to a platform. I've been using Clugo platform for enrichment analysis, but there are many web-based platforms. You can ask ChatGPT or any AI platform, or even Google can give you list of platforms that gives you options for enrichment. You just load the identifiers and you select the maybe the taxon, the species, and then you can run. But the biological insights that you want to look into, maybe you should have a reference, because sometimes if your data is coming from any cell line, and if you're looking for it, then the cell apoptosis process will be hyper or upregulated or downregulated in an abnormal level. So make sure that, I mean, these are for starters, just to remind, but this, what the question that you're asking is and an enrichment webinar, I mean, a training altogether. I think maybe IDBC can come up with a tutorial later. Yeah.
And also, I think if he want more clarity, he can always write to you, and you can give him detailed.
Yeah, maybe. Yeah, you can share my mail ID, and happy to answer questions. Yes, yes, well. Yeah.
So Purvit wants to know that how does having a standardized ontology help us compare GPCR data from this study with data from another lab?
I mean, I'm still yet to get the context because, I mean, which ontology you want to use and which subject you want to study. Because, for example, if you have proteomics data of, in GPCR, maybe plant or animal proteomics, in which space want you explore? There might be a theme. Maybe you want to explore the clinical context, or there might be you want to explore the therapeutic responses. And then you have to have a reference like before the drug usage and after the drug, and then try to compare. Again, it is another training module. Yeah, good to have these questions because we can understand the students' demand, I mean, users' demand, and then IDBC can come up with another different, series of webinars or training modules like that. Yeah.
So Aniket wants to know what is the difference between how this geo enrichment analysis is different from pathway analysis like QIAGEN reactome.
Yeah, I mean, pathway is like the Go is a category. I mean, Go finds it. It is one of the foundation one. I mean, Go enrichment. It is just a different domains. Pathway, there's a sequence of events, and Go is just that a category of the function or molecular process or the biology. So it gives you a bookmark, but then the navigation process is the entire path is the pathway. So reactome or KEGG or enrichment, it tells you where the sequence of events happen within the in cellular processes. So Go tells you, okay, the set of genes carry this function, and this will be involved in this pathway. Likewise,
Yes.
Ma'am, actually, I also wanted to know how does flow changes because of disease severity. Suppose I am working on ophthalmology, so I wanted to know the severity of diabetic retinopathy from mild to severe. So, how gene ontology will help to understand the disease severity, ma'am?
No, a gene ontology doesn't cover the clinical aspects, right? Okay, so the gene, so you have to look for monospecific ontology, but then, so no specific gene or protein is directly related to the clinical ontology. You have the designate or OMIM databases where the disease between gene to phenotype has been been established. But then your severity condition, I mean, so you need, for example, you need transcriptomics data or proteomics data in sample collected in severe patients. And then you need to have the same structure with the sample, collection with the reference, and then for the normal. So then there should be a comparison, a differential enrichment comparison going through these cases. And then you have the category mapped accordingly. The first category is your test sample, and then this sample is your reference. And how does then gradually transfer into whether this is in the beginner stage or the normal stage. I don't think any ontology can directly do that. It is based on the sequence of questions you ask and the type of data you bring into analysis in your, in your space is going to help it. Directly gene ontology cannot know. Yeah.
Okay, thank you. Thank you so much.
No
And then Arun wants to know that how does gene ontology accounts for protein isoforms?
Well, gene ontology doesn't directly connect to protein isoforms. Okay, so gene ontology, as I mentioned, the function so the gene products and its molecular function, biological process, and what else? The cellular component. The protein having multiple isoform versions doesn't affect any of the molecular function or biological process or cellular component. Maybe cellular component, maybe it can, restrict itself to the nuclear compartment or come into the cytoplasmic compartment. But beyond that, Go ontology doesn't help. But isoforms can differentially bind to protein because they have a difference in sequence length. So then they can differentially bind to proteins, and then there might be slight variation in the outcome of the process. I mean, in the biological pathway. And I have seen there are, I think, nuclear factor kappa B, maybe there are powerful isoforms which translate differential signals when any of the specific isoform has been, encouraged to be translated in the cellular process. So it depends, but there could be a change, differential binding in the protein binding, and there might be a change in the biological pathways as well, but not in terms of Go. Yeah.
So Shivam wants to know, I think he wants to know what all parameters are required for metabolomics and metabolites. Maybe I think he wanted to connect to the ontology terms. So any specific ontologies for metabolites? Or Shivam, if the question is different, you can please unmute yourself and ask the question if you are still there.
Okay, I am here, ma'am. First of all, very informative session today. Ma'am, actually, I am working on this metabolomics and for disease.
Okay.
So I want to confirm that whatever the parameters that you have told just now, we have to follow that for metabolomics as well, or it has some different parameters?
Can be different, I think.
I don't understand the parameters.
Like suppose you said, like you said just now, for genes, we have to first check the disease specific parameters, and then we have to first go for statistics as well. So in metabolomics, we are also, there is any respiratories available or not, that I want to know. Like for genes, you said.
Yeah, I don't think we have a comprehensive metadata annotated for metabolomics. Okay, so this has to be sampled, and like if you test, it is like, so we are still in the exploratory mode. I think right now the metabolomics, we have metabolites in EBI, and they have collected the metadata based on the sample, the clinical phenotype, the age, and the disease status or whatever. But I don't know if that part is available for analysis. I have checked that in Pride. I have insisted them to map the metadata ontology, which they are still doing it. And basically, there is in Pride, they have a cluster. So there was some salivary sample proteomics data, and they have compared with the regular sample data and the disease sample data as well. But all of them are, put into one link. So that doesn't tell you differentially because you need to tag these. Is this proteomics data for natural samples like regular sample, and this is for the salivary sample with the clinical condition. Similarly, I don't know if metabolomics have this metadata tagged, and unless we tag it, and then the individual metabolomics should be enabled the tag to be carried. I mean, so this is the regular level, or this is the abnormal level, or whatever the change. So that then every mapping has to be ontologically tagged so that it is available for analysis. So all the questions that you ask is reinstating the need for ontology at even the metadata mapping. Yeah.
Yes.
Yeah, thank you so much.
Okay, thank you, Shivam. Now, Isha wanted to know that how do you approach ontology selection when integrating heterogeneous data sets like phenotypic, genomic, and biodiversity data?
Yeah, but it depends on the question that you're trying to address, right? So, unless, so the Go is essentially, people mostly use Go to look, it's an exploratory phase. Like you can clusterize the genes to put them in one of the three boxes. And then if you ask me if this is a complete knowledge, well, instead of looking at the big picture, now you have boxed them. But then you should have the next question: what do you want to do with these boxes? And that will take you, if you're looking for there's a Grameen pathway database, maybe you want to study that. And then but I have, when I was discussing with Professor Shaurav Raghuvanshi, he said that there are multiple checkpoints in databases, like, in plant growth cycles. And I don't think all of them have been mapped. And we were discussing and we wanted to build something which is needed for, the multiple points that we want to study, multiple pathway points. We don't have that actually. But you have a reference, but essentially it is the type of question that you need, that you're interested to address, that defines which ontology you want to use. And then if you think that there's no such ontology, then essentially we need to build the ontology. Yeah.
Related to that, I think I have a question. Like for ICPD, we are using plantiome and crop ontology together, but
Okay.
plantiome is more of a generic ontology terms, which is universal, and I think it is based on the model plant system. And crop ontology, on the other hand, have very crop specific terms. But
Kamash, okay.
possible that when some crop specific data comes to us, and as we are dealing with phenotype data, that crop in which the user is working, it is not present on crop ontology. So we have given a functionality where he can contribute towards the creation of that ontology terms in that crop, and later we will figure out how to
Exactly.
those ontologies together.
Yeah.
So
Yeah.
so if a crop is available, and we have ontology in very crop specific ontologies also, and a generic ontology also, which we need to follow, generic one or the specific one?
Well, so I would explore both of them, but then the aspects that I want to cover, because generic ontology is like, it is common to all the plants, but then if you are looking for crop ontology, maybe there is specification that the type of, when we want to plant the plant and what is the drought specifications, there might be difference in the flavors. So I would look for which flavor that is offering, and I'll still have the generic version as my comparison preferences, and nothing can stop me exploring as much as possible. But yeah, it depends on the mood, and then more importantly, the question actually. Yeah.
So is it possible that we include both?
Of course we can. Of course we can. With the right justification, we can include both. Yes, yeah.
Okay, thank you. So then actually we have lots of questions, so people are really interested in this topic.
Well,
How does geo change across disease severity and stages? I think it again depends on different.
no, Go doesn't have that. Go, it is just telling you, it is reflecting the natural cellular process. I don't think it goes into the detail of, clinical stage. And for that, you might need to in UniProt, while you are exploring the UniProt homepage, there is a Go annotation page. Now they are including the short Go models, actually Go CAM models, causal activity models, where they add a terminology if something is inhibited in any clinical disease or phenotype. Now they're just building. So maybe Reactome could be a space where you could tell, in case of a clinical phenotype, the pathway differing and where your genes are significantly enriched within that pathway or not. But Go doesn't go into the details of clinical stage as far as I understand here.
Okay. Then Purnima wants to know, how does data curation contributes to data management in research proposal and publication? It's a generic question. I think it is relevant to all of them.
Yes. Data curation, first thing, you are doing a great benefit to the society. Okay. So it's not, it demands so much of perseverance to curate, to bring the information, knowledge locked in the publications to the mainstream, which is accessible to and so you can always say it is like, make it more open for availability, not just in the textual file, but for the analysis part, and which type of analysis, where does it fit in? Transcriptomics can be used in multiple phases. And then, yeah, and analysis, and it go for a specific pathway for a clinical disease or something like that. You can always tell that, yeah, this is, and you can give the number of visitors that you use, people who have used your portal, and if possible, collect their feedback, which they would love to see more in your database or in your portal. And that could be another supportive, point in your grant application. I mean, that's how we do. Yeah. The more the statistics, the more number of visitors you have, more engagement and more outreach activity that you do, engage people with training, people use your data, your portal for analysis. The more the information, the statistics, it's better. Yeah.
Thank you. Then Priya wants to know, is there any ontology for processes, proteins of microorganism? I think there should be.
Yeah. What on the generally for proteins? See, these Go terms cover. It's not just. The Go function. You can look for any protein using the identifier in Go ontology. It is not restricted to not just one model organism. Mostly human, but then you can still. But microbes. You can have reference model organisms. Maybe E. coli can be studied, but I don't know if specific other specific model organisms are studied. Yeah.
Then Shivangi wants to know that are there recommended workflows to map NGS derived variants onto interaction networks for pathway level analysis?
Well, that is molecular interactions, intact subject. We collect we capture the interaction evidences, the part that whenever a construct carries a genetic variant or mutation. And you can also use Cytoscape. You can build your network in Cytoscape and then add another data to be added on top of the nodes carrying the mutation. But then the mutation should also carry an effect. So then it's not just adding a mutation, then you should also add to the edge attached to it, saying that when this protein carries a mutation, and so this affects the binding of the protein, that is the protein B, and the protein B, it is attached to this edge. So that level of data that you need to have, I think Intact is already curating it. I don't know if you can externally add it to this, but you can add this as a signal. So when you have a network, so there is a network propagation model where you consider this as a signal, and then the signal gets translated within the clusters. So more the strength of the node, how much if a protein has 10 proteins attached to it, and if the parental node has a mutation and the mutation might affect the 10 proteins or not, that is calculated by an algorithm. And so that is called a propagation, network propagation application. I think you can explore in Klugo. So that can be done. And you can use Intact as a reference. Because whether the strength of the mutation can be considered as reference. You can refer to Intact and the binding, if it is broken from Intact evidence, you can say this is a crucial evidence and it cuts the interaction. So then consider this as a signal, and the signal can be translated within the network and see how far the network is broken. Because there should be a pathway the collaboration as will be associated. There'll be a sub-hub which will be associated, which might be affected badly. So that could help.
Okay. I hope Shivangi gets her answer. So now Khushbu wants to know that how can knowledge of ontology and data curation help in transitioning from dry lab to wet lab research? And what are the basic skill required to start working with biological data standards and ontologies?
The basic skill is the interest. And the perseverance to learn things. And then in terms of how to translate your wet lab research to dry lab, that is how you use your ontologies to build you have the machine code terminologies and you have the human readable terminologies, you relate them. So that is how it is processed. So when you start with curation, maybe the initial level of training, with decent training, you'll be able to use the basic mapping for cell or tissue or anything. And then looking, maybe a 6 months of browsing here and there can give you a glimpse, and you can also look for the gaps and you can sit with the experts. Like there are many academic institutions in India, but I don't think we have the setup people who are willing to volunteer for this ontology discussion part. I think that is a gap I'm trying to address. So we can sit up and whenever you have this group meetings, conferences that happen within lab group, you should always talk to the professors or anyone within the expert and then ask them like how this can be addressed. And so that this could be something that, yeah, it needs to be established, but we have not complete. As I mentioned, ontology is an ever-going process, but there are a lot of gaps that we still haven't covered, particularly in context of Indian data sets. So there's a lot of work actually.
Yes. Then Akash wants to know how ontology helps in drug discovery, virus plus host protein.
So it is just, the context that a paper carries can be translated to another data set. So that is the power of ontology. Okay, so your Google or any AI model can fetch you the concept, but the concept, unless it is mapped to the ontology, cannot be allowed to any analysis space. The commonly used analysis space is cytoscape. So everything which like you need this mapping done. So once you do the mapping, then it makes it knowledge is universal and it is available for analysis. So when a publication has information on virus infection affecting whatever a pathway, and so then we should have the information translated into the pathway database saying that because of virus infection there is an alternative change in the pathway happening, and then that has to be connected with the viral infection tag. If for example, you can go to Reactome database, which I'm suggesting that because I'm a bit more familiar with that. And so Reactome database and then choose for viral infection, there is actually a pathway that you can land into multiple pathways. And if you look for the evidence, like every pathway that you select will be coming from a publication. So if you go for it, the publication will carry the details. And if you think that the publication, there could be gaps in, not complete all the viral infection. The effect of viral infection is not completely mapped. You can go back to Reactome, ask these people to curate, find publications and ask these people to curate. That's how you engage. And then you can also learn. It is not paid, but then it gives you an experience to understand what is curation and pick up a Reactome pathway and see if you can decode the publication information into the pathway requirements for Reactome. So you can do that.
Okay. Then Isha wants to know how to handle the semantic overlap and redundancy when mapping across different ontologies.
Well, that is, you just go to the level of significance that will, because this redundancy part is just overlapping, certain areas. So increase the, if I say like in Gluco, what I do is that instead of the regular number of genes that can be allowed to map, the minimum number is 3, and I increase the number so that there should be a minimum of 5 number of genes mapped to it. Then the overlapping will be reduced. Yeah.
Nivedita wants to know what are the common mistakes researchers make when designing ontologies for data submission?
Researchers make, they don't have to, research, while data submission, research people, let us not confuse. Research people, please do the research, and then the curators handle the ontology part. And if there is any confusion, curators will communicate with the researchers and let them handle, because the overlap might sound exciting, but it is overwhelming, very demanding. I will not recommend that part unless you are an expert, double expert, triple expert, or whatever, higher level of expert. But yeah.
Then I think it's the last question of our session. How can ontology frameworks be integrated with experimental datasets from proteomics or cell line studies?
Cell line studies and proteomics experiments. So proteomics experiments already have their own metadata. And so maybe it's mapped or partially mapped, and maybe the mapping is incomplete. So at least if you want, you can choose that data set to be mapped, and when you run the analysis, you can know this proteomics data is fetched from this metadata, and this metadata you can assign a map to it, and then you can run the next set of proteomics data for your analysis, and then there will be, you can take another mapping, the differential mapping, and then this will be available for comparison. So they don't actually, I mean, there's a degree of metadata that is also mapped, but it is not complete in pride for proteomics. Cell line ontology, I don't know. Cell line ontology, I don't think there's other data type, because cell line ontology is the terminology, like, HeLa, or so that is, it is just definitive. It's a term, and then it describes what is a cell line, and then it has this identifier. I don't know what else information that can be found. I mean, it tells you it's a human, it is a cancer. Yeah.
So with this, I think we have answered all the questions, and and wonderful talk, and everybody is appreciating. Thank you so much, Kalpana.
It's
And
very engaging, very engaging.
and it's some way highlighting towards the second part of it, with directly you can show the curation and other things.
Yes.
So I think it's very nice to see that people really liked your talk.
Yeah, I like the questions, actually, because it is good to know the questions are coming in the right direction, means that we are, yeah. Okay.
So with this, I like to thank you, and thank you so much for agreeing to deliver this talk in a very short span of time. And we definitely like to welcome you again with some other interesting topic. And
Yeah, happy to connect with you. Happy to be part of this session. It's very interesting with the student training, and of course, I was not curating or doing anything for the last 2 months, but then this gave me a task to reconnect with something which I love to do. So thank you for the opportunity, and thank you for gathering, nice engagement, because this platform, I was expecting huge, but it's not that expected.
So I think we have an announcement for the participant that we will provide you the e-certificate for the participating here, and I will share the links to your email addresses, and so you will fill that feedback form, and then you can download your certificates. So I think with that, thank you again, Kalpana, for
Thank you. Thanks so
for
much.
your efforts and knowledge sharing. And with this, I think thank you all the attendees and for joining us today. And we hope that you will join us again next month with another topic. And that's how the association between IBDC and the research community of India will progress in a better direction, where we can support your research and the proper management, or you can say the fair management of your data with our data resources and knowledge sharing platforms like these webinar series. So thank you again for joining, and thank you again,
Hi.
Kalpana. Yes.
This last question. Actually, just wanted to know, can we connect with the speaker via LinkedIn or mail
Yeah.
ID?
I mean, no problem. Yeah. I mean, you can search by my name, and yeah, you
Okay,
can.
I'll just send an invite. My name is Dattadarshan. I just finished my PhD.
Okay. Yeah.
I'm interested in MBL. Actually, that's right. Just wanted to ask few questions.
Yeah. Yeah. Happy to connect with you. Yeah, we'll do that after this webinar. Yeah.
Thank you.
I was not checking any. Yeah, you can connect with me. And
Thank you.
feel free to, Sonia, feel free to share my slides with people who are interested. Yeah, no problem. Yeah.
Actually, this webinar is recorded, so we will
Okay.
put this recording on IBDC YouTube channel, so
Yeah.
people to see this whenever they want, and you can connect all of you can connect with her on LinkedIn. She's there on LinkedIn, and you can get more knowledge from there. So thank you again for joining us today. And with this, I think, thank you so much, and goodbye for now.
Thank you, Sonia, for the opportunity, and thank you, students, for the very engaging session. And we might connect in some other platform, but until then, bye. Thank you so much.
Bye, everyone. Thank you.
I'm leaving. Yeah.